At 200 meters deep, the sun disappears. Not dims, disappears. Below that point, there is no photosynthesis, no color as the human eye perceives it, no visual reference for up or down, and no creature that looks anything like what lives above. The ocean does not get gradually stranger as you descend. It gets categorically stranger, in steps, each layer defined by a new set of physical conditions that have produced a new set of biological solutions that nothing on land and nothing in the layer above could have predicted. This is a journey through those layers, from the familiar to the improbable to the flatly impossible by any biological intuition that the surface world provides. Every 100 meters down is a different planet. And by the time you reach the bottom, you will no longer be surprised that science keeps finding things it does not have names for. If you enjoy drifting into the deep ocean and discovering what lies beneath, please consider liking and subscribing. It truly helps more than you know. Are you ready? Let's begin. The ocean you think you know is only the top 200 meters of it. Everything you have seen in photographs, everything you have swum through on a vacation, everything that appears in wildlife documentaries filmed with natural light, everything that feels familiar and navigable about the sea fits inside a layer that represents roughly 2% of the ocean's total depth. 2%. The other 98% lies below the point where usable sunlight penetrates, and it begins right at the edge of what most people would consider a deep dive. This fact does not register the way it should, because the surface of the ocean looks like the ocean, and the ocean looks like one thing rather than a stack of completely different worlds layered on top of each other, each one darker and colder and stranger than the one above it. But that is what it is, a stack of worlds. And the further down you go into that stack, the more the creatures living there begin to look like something that evolution should not have been able to produce, something that tests the limits of what biology is even supposed to be capable of. This is where the story starts, at the top, in the light, with the things that seem familiar, that look like fish and sharks and whales in ways the eye can parse and the mind can accept. Because the familiar is where you need to begin before what comes next can land the way it should. The sunlight zone extends from the surface to a depth of approximately 200 meters, and it is the only part of the ocean where sunlight penetrates in quantities sufficient to drive photosynthesis. Everything alive in the ocean ultimately depends on this layer, because the phytoplankton that converts sunlight into organic matter at this depth are the foundation of nearly every marine food chain that exists. From the microscopic copepods that graze on them, to the blue whales that travel thousands of kilometers to exploit the regions where plankton blooms are most dense. The sunlight zone is where primary production happens, where the sun's energy enters the ocean food web, and where the biological activity per unit of water is higher than anywhere else in the ocean's volume. It is also the most studied, most observed, and most familiar part of the ocean, to the point where it can be easy to assume that familiarity with this layer constitutes knowledge of the ocean as a whole. It does not. It constitutes knowledge of the lobby. The great white shark is the animal most people would choose as the emblematic predator of this layer, and the great white's biology is instructive precisely because even in the most familiar, most studied, most photogenic marine predator alive, the actual biology turns out to be stranger than the popular image suggests. The great white is partially endothermic, meaning it can maintain the temperature of certain body regions above that of the surrounding water, through a system of countercurrent heat exchange in which warm blood traveling away from the swimming muscles transfers heat to cool blood, returning from the extremities before that cool blood can lower the temperature of the core. This regional endothermy allows the great white to function effectively in water temperatures that will leave most sharks sluggish and slow, giving it access to cold, productive coastal waters, where pinniped colonies concentrate, and where prey is available in densities that the shark's metabolic demands require. The great white is not simply a cold-blooded killing machine, the phrase that popular accounts use almost without exception. It is a physiologically sophisticated animal doing something that most sharks cannot do, maintaining warm muscles in cold water, which is why it can hunt effectively in environments as different as South Africa, California, and southern Australia. The popular image of the great white as a simple, prehistoric predator driven purely by instinct is not supported by the biology. It is a highly adapted, cognitively capable apex predator doing something physiologically complex that most of its relatives have never managed. The animal you think you know is not the animal that is actually out there. The octopus is the animal in the sunlight zone whose biology most thoroughly defies the expectations that familiarity with it seems to create. Most people know that octopuses can change color and texture. Most people assume this is a visual process, that the octopus sees the background it wants to match and adjusts its skin accordingly. The biology is more interesting and more mysterious than this. Octopuses are colorblind. Every known species of octopus has only a single type of photoreceptor in its eyes, which means it cannot distinguish wavelengths of light the way that animals with color vision can, which means it cannot see the colors it is producing in its own skin. It is matching colors it cannot see to patterns it perceives only in terms of brightness and contrast, with a precision that has routinely fooled predators with full color vision for millions of years. How it does this has not been fully resolved. One hypothesis, supported by the discovery of photoreceptive proteins in octopus skin cells, is that the skin itself may be detecting color directly, independent of the eyes, allowing the animal to perform a kind of distributed color sensing across its entire body surface rather than relying on centralized visual processing. This would represent a sensory architecture that has no parallel in vertebrate biology and that the research community is still actively investigating. The octopus may be seeing with its skin in a way that no optical instrument and no visual system science has previously described, and the mechanism by which it achieves its camouflage may involve sensory biology that current scientific frameworks are not yet fully equipped to characterize. The color spectrum itself begins to disappear in the sunlight zone before the light does, and the sequence in which colors vanish with depth is itself a piece of evidence about the physics of water and the biology that has evolved around it. Red light has the longest wavelength in the visible spectrum and is absorbed by water most rapidly, disappearing within the first 10 meters. Orange follows, then yellow, then green, until at 200 meters, in the clearest ocean water, only the faintest blue remains before that too fades to nothing. This is why deep-sea photographs taken without artificial light show everything in shades of blue and gray, and why the blood of a fish at depth, which is red at the surface, appears green or black at the depth where red wavelengths no longer exist. Animals that have evolved in these intermediate depths have taken advantage of this color physics in ways that blur the line between camouflage and invisibility. Many midwater fish and crustaceans are red or deep orange, colors that at the surface would make them immediately visible, but that at depth, where red wavelengths have been absorbed, render them as dark and invisible as black against a dim blue background. Evolution found the loophole in the physics of light absorption and produced animals that are simultaneously colorful in the air and invisible in the water where they actually live. The bioluminescence that appears in the sunlight zone is a preview of a phenomenon that becomes progressively more dominant and more biologically significant as depth increases, and its presence near the surface illustrates how widespread the production of biological light actually is across marine life at all depths. Dinoflagellates, single-celled plankton that produce brief flashes of blue light when mechanically disturbed, are responsible for the glowing waves and sparkling wakes that anyone who has swum in the ocean at night in certain locations has seen. The light they produce is not decoration. It is a defense mechanism, a burglar alarm light that, when triggered by the movement of a predator trying to eat the dinoflagellate, illuminates the predator and makes it visible to its own predators. The dinoflagellate cannot outswim a copepod trying to eat it, but it can make the copepod visible to the fish trying to eat the copepod, recruiting a higher-level predator as an involuntary bodyguard through the medium of light. The dinoflagellate cannot outswim what is trying to eat it, but it can make what is trying to eat it visible to something larger. It is weaponizing light it produces in its own body to recruit a predator it has never met to defend it from a threat it cannot see. In the sunlight zone, even the smallest organisms are playing a game more sophisticated than it looks. Every single day, at dusk, the largest animal migration on Earth happens, and almost nobody knows it is occurring. Billions of organisms that have spent the daylight hours hiding in the darker, safer water below 200 meters rise toward the surface to feed under the cover of darkness, and descend again before dawn to avoid the visual predators that hunt in daylight. The total biomass involved, integrated across the entire global ocean, is estimated in the billions of tons. This migration happens every night, everywhere, simultaneously, without pause, in an ocean that, from the surface, looks like it is simply sitting still. The first time Navy sonar operators detected this layer rising at dusk and descending at dawn, they thought they had found a second seafloor, a false bottom that moved. It took years to determine that what they were tracking was not geology, it was life. The largest single movement of animals on this planet, and it had been happening every day for millions of years before anyone had an instrument sensitive enough to notice it. The sensory biology of sharks in the sunlight zone illustrates how the familiar surface ocean contains detection systems that operate on physical principles entirely outside human sensory experience. The ampullae of Lorenzini gel-filled pores on the snout of sharks detect the weak electrical fields produced by the muscle contractions and nerve impulses of other animals, allowing sharks to locate prey that is invisible, motionless, and buried in sediment purely by the electrical signature of its living tissue. The sensitivity of this system can detect electric fields as weak as 5 billionths of a volt per centimeter, the most acute electroreceptive sensitivity measured in any animal. A shark, in the final approach to concealed prey, is navigating by a sense that no human organ possesses, in a dimension of the physical world that is entirely invisible to human experience, but that for the shark is as rich and specific as vision is for us. The sunlight zone is also where one of the ocean's most consequential and least visible weapons exists: venom. Box jellyfish, present in tropical coastal waters, carry tentacles lined with nematocysts, microscopic harpoon-like structures that fire on contact and inject one of the most potent venoms produced by any animal. A large box jellyfish has enough venom to stop a human heart in minutes. The animal itself is almost completely transparent, produces no bioluminescence, has no skeleton, and can contract its bell to move at speeds that make it one of the fastest jellyfish alive. It has 24 eyes arranged in 4 clusters, and some of these eyes form clear images in a way that allows the jellyfish to navigate around objects rather than drifting passively. A box jellyfish sees the world. It has no brain. The visual processing that tells it where to go happens in a nerve net distributed through its body rather than in any centralized structure. It is seeing without a brain to see with, navigating without a mind to navigate with, and killing with a weapon so small it is invisible to the naked eye. This is the sunlight zone. This is the part of the ocean that feels familiar. The familiar creature of the sunlight zone that best previews what becomes increasingly extreme in the layers below is not the great white shark, or the whale shark, or the octopus, but the common dolphin. The dolphin is a mammal that returned to the sea from land approximately 50 million years ago, and in the process of returning, it rebuilt its body plan, its sensory systems, and its cognitive architecture for an aquatic existence while retaining the warm blood, air-breathing, and live birth of its terrestrial ancestors. The echolocation system that dolphins use to navigate and hunt in water, producing clicks that travel through the water, reflect off objects, and return to receptors in the dolphin's lower jaw and inner ear as acoustic images of the surrounding environment, is a sensory technology that has no vertebrate equivalent outside of bats and odontocet cetaceans, and that the dolphin's nervous system has developed into a tool of extraordinary precision. A dolphin can detect a fish the size of a tennis ball at ranges of 100 meters through its echolocation, can determine the density and internal structure of objects it has never encountered before, and can share acoustic information with other dolphins in ways that suggest the echolocation signal contains more information than the dolphin simply needs for its own navigation. The dolphin in the sunlight zone is using physics that humans have only recently learned to exploit technologically, and doing so with a biological instrument that has been refined by 50 million years of evolution in the ocean. It is, in the sunlight zone, one of the less strange things alive. What comes below is considerably less familiar, and the strangeness increases with every meter of depth in a progression that follows no gradient more reliable than the one that leads away from the light. At 200 meters, something fundamental changes, not gradually. The transition from the sunlight zone to the twilight zone, the mesopelagic, is not a slow dimming of the kind that happens at dusk on land, where the eye adjusts incrementally and the world goes from bright to dark over the course of an hour. In the ocean, the change is faster and more absolute. Below 200 meters, the light that remains is not enough for photosynthesis. It is not enough to cast a shadow. It is a faint, directionless blue glow that exists more as a memory of the surface than as usable illumination. And below 400 to 600 meters, even that is gone. The twilight zone runs from 200 to 1,000 meters and is the layer where the ocean begins to reveal what it has been keeping from the surface world. The creatures here have had millions of years to adapt to conditions that include near total darkness, water temperatures between 4 and 15 degrees Celsius, and pressures ranging from 20 to 100 atmospheres. What they have produced in response to those conditions is a collection of body plans and biological strategies that test the boundaries of what animal life is supposed to look like. The viperfish is one of the first animals encountered in the twilight zone that requires a genuine rethinking of what a fish can be. It reaches a maximum length of approximately 30 centimeters, which makes it a small fish by any surface standard, but its proportions bear no relationship to anything that lives in the sunlight zone. The fan-like teeth that give it its name are not merely large relative to its body. They are so long that they cannot fit inside a closed mouth, which means the viperfish swims with its jaws permanently open, its lower teeth projecting forward and upward like a set of transparent glass needles that can be seen through the fish's translucent body. The teeth have a specific function beyond simple prey capture. When the viperfish lunges at prey, the long fangs impale the target before the mouth fully closes, preventing escape in the fraction of a second before the jaw engages. This is not the strike geometry of a surface predator. It is a strike geometry designed for a specific problem that the twilight zone presents. The problem of hunting in near total darkness, where reaction time is the limiting factor and any prey item that escapes cannot be followed effectively in three-dimensional dark water. The viperfish solution is to ensure that the prey is already impaled before it knows what has happened. Below the fang structure, the viperfish has a light-producing organ along its dorsal fin that dangles above its head like a lure when it swims. The strategy is complete. Attract with light, impale with glass needles before the prey can react, swallow with a jaw that unhinges to consume animals larger than the viperfish itself. It is not a predator in the way surface predators are predators. It is a mechanism refined by millions of years of darkness into something that functions more like a trap than an animal. The hatchetfish represent a different and equally remarkable solution to the problem of visibility in the twilight zone. Their name comes from their extreme lateral compression, the body flattened from side to side so severely that the fish is almost two-dimensional when viewed from the front, with a deep keel along the ventral surface and a body plan that looks more like a piece of machined metal than a living animal. The function of this shape is camouflage of a specific and counterintuitive kind. In the twilight zone, the faint light comes from above, filtering down from the surface. Any object in the water column casts a silhouette when viewed from below, a dark shape against the slightly less dark background of the filtered surface light. Predators hunting from below look up and search for these silhouettes. The hatchetfish's extreme flattening minimizes its silhouette from below, making it nearly invisible when viewed directly from underneath. But the silhouette, however minimal, would still be visible against the background light. The hatchetfish has solved this problem with bioluminescence. Its ventral surface is lined with photophores that produce downward-directed light, matching the intensity and color of the ambient light from above. This counter-illumination means that when viewed from below, the hatchetfish produces exactly as much light as the background behind it, making it effectively invisible against the faint surface glow. The hatchetfish is not camouflaged against the seafloor or the water column. It is camouflaged against light itself, matching the photon output of its own body to the photon output of the filtered sun above it. The counter-illumination strategy of the hatchetfish requires a sensory system capable of measuring the ambient light intensity from above and adjusting the photophore output to match it in real time. As the fish moves through water layers of varying transparency, and as cloud cover and time of day change the intensity of the surface light being filtered down, this regulation is not something the fish consciously performs. It is handled by a biological feedback system that continuously samples the ambient light and adjusts the photophore output accordingly. A kind of living dimmer switch operating across dozens of individual light organs simultaneously. The precision required for this matching is such that measurements of hatchetfish bioluminescence in the wild have found their ventral light output matching the background light intensity to within a factor of 2 or less, close enough to render the fish effectively invisible to predators whose visual systems are adapted to the contrast ratios of the twilight zone. The hatchetfish is not merely hiding in the dark. It is solving a continuous differential equation in light physics with its own body. The oarfish is not a twilight zone permanent resident in the way that the viperfish and hatchetfish are, but it moves through the upper twilight zone and has been observed at depths below 200 meters, frequently enough to be considered a mesopelagic species for much of its life. It is the longest bony fish alive, documented at lengths exceeding 11 meters and credibly reported at lengths approaching 17 meters in some historical accounts, which means it is a fish longer than a city bus that most people have never seen and that science had never photographed alive in its natural habitat until 2001. The oarfish's body plan is bizarre in a way that defies easy summary. It is extremely laterally compressed, ribbon-like, with a dramatic dorsal fin running the full length of its body composed of hundreds of individual fin rays, each tipped with a small, fleshy extension that creates a fringe effect along the top of the animal. Its pelvic fins are reduced to long, trailing filaments that end in paddle-shaped structures. It appears to swim primarily by undulating this dorsal fin, rather than by the body movements used by most fish, holding itself nearly vertical in the water column with its head up, drifting through the twilight zone in a posture that no other fish adopts. The specific reason for this vertical orientation whether it is a feeding strategy, a behavioral response to some aspect of the twilight zone's physics, or a posture that serves a social or reproductive function, is not fully established. The oarfish is so rarely encountered alive in its natural habitat that most of what science knows about its behavior comes from animals that were already dying when they were found. A fish longer than a school bus, and the only ones science has ever seen were the ones that did not make it back down. The deep scattering layer is the defining biological phenomenon of the twilight zone and the feature that most clearly illustrates how much biological activity is happening in this part of the ocean that the surface world never sees. The deep scattering layer was first detected during World War II when naval sonar operators noticed that their echo sounders were returning signals from an apparent seafloor that was located hundreds of meters above the actual seafloor, and that this false bottom rose toward the surface at night and descended during the day. The false bottom was, as eventually determined, a layer of living organisms so densely packed that their bodies collectively reflected sonar signals in the same way the seafloor did. The depth to which sonar signals penetrated before being reflected back told operators not how deep the water was, but how deep the layer of organisms was, and the layer's daily movement from depth toward the surface and back again at dawn reflected the daily vertical migration, the same behavior observed at the boundary of the sunlight zone, but driven by organisms living throughout the twilight zone's water column. The layer is primarily composed of fish, with gas-filled swim bladders, squid, and deep-sea crustaceans, all packed densely enough that their bodies collectively reflect sonar signals the way a solid surface does. The Navy operators who first detected it were not looking for fish, they were looking for submarines. What they found instead was the largest concentration of animal life on Earth, hiding in plain sight a few hundred meters below every ship that had ever crossed the ocean, entirely invisible until the sonar looked directly at it. The predators that hunt in the twilight zone are shaped by the same conditions that shape their prey, and several of them represent evolutionary solutions to predation in near darkness that are as unusual as the defensive adaptations of the animals they hunt. The dragonfish include the viperfish and several related species that have independently evolved the combination of bioluminescent lures, extensible jaws, and transparent fan-like teeth that characterize Twilight Zone ambush predators. Some dragonfish species have taken the bioluminescent strategy one step further, evolving photophores below their eyes that produce red light rather than the blue light produced by most deep-sea bioluminescent organisms. This matters because most deep-sea animals, including the prey species that dragonfish hunt, are sensitive only to blue light and cannot detect red wavelengths at all, effectively rendering red light invisible to them. A dragonfish using its red photophores to illuminate its visual field is essentially carrying an infrared torch in a world where nothing else can see infrared. It can see its prey clearly, while the prey is functionally blind to the light being used to find it. The sperm whale is the most dramatic visitor to the Twilight Zone from the surface world, diving through the mesopelagic to reach the prey that drives its biology. Sperm whales feed primarily on squid, many of them mesopelagic species, and their dives through the twilight zone on the way to the deeper water, where their largest prey concentrates, represent regular transits through a layer whose conditions would be immediately lethal to any unprotected human. A sperm whale diving through the twilight zone is holding its breath, its lungs collapsed under pressure, its heart rate reduced to a fraction of the surface rate, its blood oxygen reserves being carefully managed by a physiology refined over millions of years for exactly this purpose. It can detect the squid it is hunting through echolocation clicks that travel through the dark water and return as acoustic images detailed enough to identify species, size, and trajectory at ranges of hundreds of meters. The sperm whale in the twilight zone is using sonar more sensitive and more precise than anything the warships that accidentally discovered the deep scattering layer could produce. Hunting prey in conditions where the human-designed instruments that first detected the layer's existence could not resolve individual organisms at all. The gelatinous organisms that drift through the twilight zone without any of the active adaptations of the dragonfish or the hatchetfish or the sperm whale are perhaps the most alien things alive in this layer, and the least studied. Siphonophores, colonial organisms that are technically collections of individual zooids performing different functions within a shared body plan, can reach lengths of 40 meters or more in the twilight zone, making them the longest animals on Earth by any standard measurement. A single siphonophore 40 meters long is not a single organism in the conventional sense. It is a colony of hundreds to thousands of individual animals, each one specialized for a specific function: swimming, feeding, reproduction, defense, that together form a structure that behaves as a coordinated whole despite being composed of genetically identical but structurally distinct individuals. The longest animal on Earth is not a whale. It is a colonial organism that looks like a thread of glass beads drifting in the dark that most people have never heard of, and that science still does not fully understand in terms of how its component individuals coordinate their behavior to produce the responses of the colony as a whole. The bioluminescence of the twilight zone is not simply more of the same phenomenon that appears near the surface in the form of glowing waves and dinoflagellate flashes. It is a qualitatively different biological system, more diverse, more functionally specialized, and more deeply integrated into the ecology of the layer than surface bioluminescence ever approaches. Studies conducted using blue water diving, a technique in which researchers descend into the open ocean water column at night without a submersible, relying only on their own diving equipment, have found that nearly every organism encountered in the upper twilight zone produces light of some kind at some point in response to some stimulus. The light is not random. Different species produce light of different colors, different intensities, different flash rates, and different patterns. And these differences appear to carry specific information, whether warnings to predators, lures for prey, or signals to conspecifics, that varies with the species and the context. The total bioluminescent communication occurring in the twilight zone at any given moment across billions of organisms simultaneously producing and responding to light signals in a layer of near-total darkness represents a form of biological information exchange whose full complexity has never been measured and likely exceeds anything that current monitoring technology could capture comprehensively. The twilight zone is not dark. It is full of light that human eyes were not built to see, and that science has only recently developed the tools to begin observing directly. The pressure at the base of the twilight zone, 1,000 meters depth, is approximately 100 atmospheres. A figure that means every square centimeter of any surface at that depth is experiencing the equivalent of 100 kilograms of force pressing against it continuously. The organisms living at this depth are not merely tolerating this pressure, they are built for it, with cellular membranes tuned to remain fluid rather than rigid at these pressures, with proteins stabilized against the tendency to change shape under compression that destroys the function of proteins adapted to surface conditions, with the very molecular architecture of their biology calibrated to conditions that would destroy the molecular architecture of any surface organism placed in them without adaptation. The 1,000-meter mark is not simply a depth that some organisms happen to survive. It is the lower boundary of a biological layer where the pressure has reshaped life itself, and what waits below it in the midnight zone represents not just more of the same adaptation, but an entirely new category of biological existence. Below 1,000 meters, the last trace of surface light is gone, not diminished, gone. There is no blue glow, no faint directional gradient to indicate where the surface is, no photons from the sun reaching this depth under any conditions at any time of year anywhere on Earth. The midnight zone, the bathypelagic, runs from 1,000 to 4,000 meters, and it is a place where the physical conditions have moved so far from anything the surface world prepares you for, that the biology here can no longer be described as variations on familiar themes. It has to be described on its own terms, as a set of solutions to a set of problems that do not exist anywhere else, produced by evolution over timescales long enough to arrive at answers that look, from the surface, like they should not be possible. The pressure here ranges from 100 to 400 atmospheres. The temperature is between 1 and 4 degrees Celsius throughout the layer, cold enough that metabolic rates run at a small fraction of what they would be in the warm surface ocean, which is itself one of the keys to understanding how organisms this deep manage their energy budgets in an environment where food is extraordinarily scarce. The midnight zone receives almost none of the organic material produced by photosynthesis in the sunlit surface. What food exists here sinks from above, in the form of dead organisms, fecal matter, and the bodies of animals that have migrated down from shallower water and stopped functioning there. The midnight zone is fed by the surface world's leftovers, and its inhabitants have evolved to survive on that. The anglerfish is the animal of the midnight zone that appears most often in popular descriptions of the deep sea, and with good reason. The bioluminescent lure that dangles from the top of its head, the esca, a modified dorsal fin ray packed with light-producing bacteria that glow with a blue-green light in the absolute darkness of the bathypelagic, is one of the most elegant evolutionary solutions to the problem of predation without light that biology has ever produced. The anglerfish does not hunt, it waits. It floats, nearly motionless, in the darkness, its lure pulsing with light, until something approaches the light and comes within range of its jaws. The jaws then close around the prey in a strike so fast that high-speed video footage struggles to capture it, driven by a mechanism so effective that the anglerfish can consume prey larger than its own body, its stomach stretching to accommodate prey items that surface predators would be physically unable to swallow. The teeth of the anglerfish angle backward, toward the throat, which means that anything that enters the mouth continues moving toward the stomach regardless of what efforts it makes to escape. Pulled along by the backward-pointing teeth with every struggling movement, the anglerfish is a trap as much as it is a predator. But the most extraordinary thing about the anglerfish is not the lure or the jaws. It is the reproductive biology of the deep-sea species, which represents one of the strangest evolutionary outcomes in vertebrate biology. The deep-sea anglerfish that most people picture, the grotesque, fang-toothed, bioluminescent predator with a light dangling from its head, is always a female. The male deep-sea anglerfish is a fundamentally different animal. It is tiny, typically a few centimeters long, compared to the female's potential size of 30 to 50 centimeters or more. It has no lure, no elongated teeth, no stomach capable of stretching to accommodate large prey. What it has in some species are enormously developed sensory organs, large eyes and large nostrils, tuned to the specific chemical and light signals produced by females of its species, because the male's entire biological purpose, from the moment it achieves reproductive maturity as a juvenile, is to find a female and attach to her. When it does, in the species that carry this strategy to its extreme, it bites into her flesh and does not let go. Over the following weeks and months, the tissues of the male and female fuse at the attachment site. The male's circulatory system connects to the female's. His eyes degenerate. His internal organs, no longer needed for independent survival, atrophy. His body is progressively absorbed by hers until what remains is a small nodule of male tissue permanently attached to the female's body, connected to her bloodstream, receiving nutrition from her metabolism, and releasing sperm when her reproductive hormones signal that spawning is occurring. The male anglerfish does not simply mate with the female. He becomes part of her, a permanently attached reproductive appendage that the female carries for the rest of her life. This strategy, called sexual parasitism, is the most extreme form of sexual dimorphism known in vertebrates. A female anglerfish can carry multiple males attached to her body simultaneously. The males, once attached and fused, are no longer independent organisms in any meaningful biological sense. They are, depending on your definitions, either parasites, organs, or mates, and the difficulty of placing them in any of those categories cleanly reflects the genuine biological novelty of what the anglerfish has done with the problem of finding a mate in complete darkness in an environment where two individuals of the same species may never encounter each other at all, if they do not have a reliable mechanism for locating each other across the vast, empty, dark water of the bathypelagic zone. The anglerfish solved the encounter problem by making the encounter permanent. Once found, the mate is never lost again. The vampire squid occupies a position in the midnight zone's biology that illustrates how the deep ocean can preserve evolutionary lineages that have no living relatives anywhere else. It is neither a true squid nor a true octopus. It belongs to its own taxonomic order, one that diverged from the ancestors of both squid and octopus approximately 300 million years ago, and has no other surviving members. It is, in the clearest sense possible, a living fossil, a representative of a lineage that existed before the age of dinosaurs and that has persisted to the present in an environment so stable and so isolated from the ecological changes of the surface world that it has had no pressure to evolve dramatically from the form it had hundreds of millions of years ago. The vampire squid lives in the oxygen minimum zone, the layer of the ocean at roughly 600 to 900 meters, where oxygen concentrations drop to levels that are lethally low for most animals, because decomposing organic matter consumes oxygen faster than circulation replenishes. The vampire squid, thrives there. Its blood contains a form of hemocyanin with an unusually high affinity for oxygen at low concentrations, allowing it to extract the maximum possible amount of oxygen from water that most animals cannot breathe at all. It has the lowest metabolic rate ever measured for a cephalopod. It moves slowly, conserves energy obsessively, and feeds not by hunting live prey, but by collecting marine snow, the constant gentle rain of dead organic particles that sinks from the productive surface layers, using sticky filaments that trail from its arms to gather these particles and transfer them to its mouth. The vampire squid is not a predator. It is a collector, passively harvesting the leftovers of the surface world's productivity in an environment where almost nothing else can survive. The fang-toothed fish holds the distinction of having the largest teeth relative to body size of any fish known to science. In a fish that reaches a maximum length of approximately 16 centimeters, the fangs are so large that they cannot be accommodated inside a closed mouth even by folding into special sockets. And the lower fangs, when the mouth is closed, actually pass outside the fish's head and sit in corresponding sheaths alongside the brain. The visual impact of the fangtooth, when encountered in ROV footage, is one of pure biological improbability. It looks like something produced by imagination rather than evolution, a cartoon monster rather than a real organism with a functional biology. But the proportions are entirely functional. The fangtooth lives in the midnight zone, where encounters between predator and prey are rare and unpredictable, and where any prey item that presents itself must be secured immediately and completely, with no possibility of it escaping and no reasonable expectation of another meal arriving soon. The enormous teeth ensure that anything seized is not released. The economy of the midnight zone does not allow for misses. The giant squid passed through the midnight zone on the relatively rare occasions that it descended below its usual depth range in the mesopelagic and bathypelagic, and the history of science's encounter with this animal is itself a story about how the midnight zone keeps its secrets. Giant squid were known for centuries, from washed-up carcasses, from the remains found in sperm whale stomachs, and from tentacles and arm fragments recovered by surface vessels. But no living individual was ever photographed in its natural habitat until 2004. and no footage of a living giant squid in the deep ocean was obtained until 2012. For 150 years after the species was formally described by science, the largest invertebrate on Earth was known entirely from dead specimens. The animal that was responsible for the sucker scars on the skin of sperm whales, evidence of genuine battles between the two largest invertebrate and vertebrate predators in the ocean, was invisible. It was leaving evidence of its existence everywhere, in the form of beaks in whale stomachs and scars on whale skin, but no human instrument had ever seen it alive. The midnight zone can hide an animal the size of a school bus for a century and a half without any particular effort. The bioluminescence of the midnight zone reaches its maximum ecological significance here, where it is the only light that ever exists. In the sunlight zone, bioluminescence is a supplementary communication channel alongside vision in ambient light. In the twilight zone, it supplements the fading filtered light from above. In the midnight zone, it is everything. Every visual communication that occurs between organisms, every predator that uses light to hunt, every prey animal that uses light in defense, every individual that uses light to signal its species or reproductive status to potential mates, operates entirely in the medium of self-produced bioluminescence against a background of absolute darkness. The eyes of midnight zone animals have been modified accordingly, with extremely large pupils, high concentrations of rod photoreceptors sensitive to very low light levels, and, in some species, tubular eyes that maximize light collection by pointing forward rather than to the sides, sacrificing the wide field of view that surface animals require for the narrow, forward-directed light amplification that the midnight zone demands. Some midnight zone fish have downward-pointing tubular eyes, specifically oriented to detect the silhouettes of prey above them against the faint upwelling bioluminescence of organisms below, inverting the visual geometry that surface predators use. The midnight zone has inverted not just the direction from which light comes, but the visual architecture that hunters use to find their prey within it. The economy of the midnight zone runs on scarcity in ways that shape everything about the animals living in it. A meal here might be a week away, a month away, or never. The organisms that survive are not the fastest or the most aggressive. They are the most patient, the most efficient, and the most capable of waiting. A fangtooth can go months without eating. A vampire squid drifts rather than swims, spending as little energy as possible for as long as possible. The anglerfish does not chase. It hangs in the darkness and lets the darkness do the work. Every adaptation in the midnight zone is, at its core, an answer to the same question. How do you stay alive in a place where almost nothing edible exists? The answers evolution has produced here are stranger than anything the surface world has needed to invent. The pressure in the midnight zone is the other architect of its biology, and it operates at the molecular level in ways that surface biology gives no preparation for. At 4,000 meters, the lower boundary of the zone, the pressure exceeds 400 atmospheres. At these pressures, water behaves differently than it does at the surface. Its viscosity increases, slowing the chemical reactions that cells depend on. The proteins of any surface organism brought to this depth would begin to change shape, losing the precise three-dimensional structure that makes them functional, becoming useless strands of amino acids within minutes. The organisms of the midnight zone carry, in the structure of every protein they produce, a molecular modification that prevents this from happening. Their enzymes are pressure-stable versions of enzymes that appear in surface life, subtly altered at specific positions in their structure to resist the crushing that 400 atmospheres imposes. This is not a small adaptation. It is a complete rewrite of the molecular machinery of life, performed over millions of years, invisible to any examination that does not go down to the atomic level of protein architecture. When deep-sea organisms are brought to the surface, this adaptation works against them. Their pressure-stable proteins malfunction at surface pressure in the same way surface proteins malfunction under theirs. They're not adapted to the deep sea, they're made for it. Surface is the alien environment. The midnight zone has shaped not just the bodies of the animals that live in it, but every strategy they use to survive it. Finding food in the dark, finding a mate in the dark, finding each other at all in a space so vast and so empty that the animals living in it have invented entirely new categories of biology to solve problems that surface life has never had to face. Every adaptation described here, the anglerfish's parasitic marriage, the fangtooth's economy of violence, the giant squid hiding from science for a century and a half, represents a solution to a problem that does not exist anywhere else. And at 4,000 meters, the midnight zone ends, the abyss begins, and the problems get harder. At 4,000 meters, the water column gives way to the abyssal zone, and the word abyssal is not an exaggeration. It comes from the Greek word for bottomless, and while the abyss does have a bottom, the word captures something true about what it feels like to descend into it, conceptually, if not literally. The abyssal zone runs from 4,000 to 6,000 meters and covers more of Earth's surface than all the continents combined. It is the dominant environment on this planet by area, the largest single habitat that exists, and it is also one of the least studied, least visited, and least understood environments anywhere in the known solar system. The conditions here are consistent in a way that the zones above are not. The temperature stays within a narrow range of 1 to 3 degrees Celsius throughout the zone. The pressure ranges from 400 to 600 atmospheres, and the darkness is total and absolute. There are no seasons in the abyssal zone. There is no day and no night. There is no variation in light because there is no light. The only variation in the environment comes from the slow, irregular rain of organic particles from above, and from the occasional geological events, earthquakes, turbidity currents, and hydrothermal activity that disturb the otherwise featureless plains of fine sediment that cover most of the abyssal seafloor. Into this environment, evolution has sent organisms that look less like the familiar animals of the surface world than anything that has yet been described. And some of the adaptations they have developed here exist nowhere else in the known biosphere. The xenophyophores are the largest individual cells known to exist anywhere on Earth, and they live on the abyssal seafloor. A single xenophyophore can reach 20 centimeters in diameter and is visible to the naked eye, which is extraordinary for a single cell, because the biology of cells at the scale that humans encounter them operates under strict physical constraints that prevent individual cells from reaching visible size without specialized solutions to the problems of nutrient transport, waste removal, and structural support that multicellular organisms solve by dividing those functions among many cooperating cells. The xenophyophore solves these problems by incorporating sediment particles from the seafloor into its structure, building a mineral skeleton of foreign material that provides the rigid support that allows the cell to maintain its enormous size without collapsing under its own cytoplasm. Inside this mineral framework, the cell's cytoplasm, the living material of the single cell, is distributed in a network of tubes and chambers that maximizes the surface area available for exchange with the surrounding water. The xenophyophore is not simply a large cell sitting on the seafloor. It is an engineered structure built by a single cell using materials it collects from its environment, whose architecture solves engineering problems that multicellular organisms solved by evolving a completely different organizational strategy. The largest individual cell on Earth builds its own skeleton from the sediment of the ocean floor, grows to the size of a human fist without dividing, and does so in one of the most inhospitable environments on the planet. Cell biology does not yet have a complete explanation for how this is possible. The abyss does it anyway. The transparency that many abyssal organisms have evolved represents a different approach to invisibility than the counter-illumination of hatchetfish or the camouflage patterns of surface animals, and it illustrates how the total absence of ambient light changes the fundamental logic of visual predator-prey interactions. In the abyssal zone, where the only light is bioluminescent and therefore directional, produced by individual organisms rather than diffused through the water column from above, a transparent body is nearly invisible from almost any angle because there is no ambient light to refract or scatter off the body's surfaces in ways that would reveal its presence. A glass squid, a comb jelly, or a transparent eel larva in the abyssal zone carries its organs in a body that reveals nothing of its presence to any visual predator that does not happen to be producing bioluminescence from a direction that creates a specific optical effect. Even in that case, what is visible is the dark, pigmented gut contents and other internal organs rather than the body as a whole. A minimal visual signature compared to the opaque body of a surface animal that reflects ambient light from every surface. Transparency in the abyssal zone is not a novelty. It is a fundamental design principle adopted independently by dozens of unrelated lineages that have all converged on the same solution to the same problem, being a body that light passes through rather than reflecting from. The glass octopus is perhaps the most visually striking example of abyssal transparency. A cephalopod whose body is so thoroughly transparent that its internal organs are visible from outside. The eyes and digestive tract and reproductive organs clearly defined within a body that otherwise appears to be made of nothing but lightly tinted water. The glass octopus was known from specimens recovered by trawl, which typically resulted in damaged and collapsed tissue that gave little impression of the animal in life, until recent ROV footage began capturing living individuals in the water column and revealed what the animal actually looks like when intact. What the footage showed was something that appeared less like an animal and more like a collection of organs suspended in transparent gel, the components of a cephalopod biology, visible through a body that contributed no visual presence of its own. The glass octopus hunts bioluminescent prey, and the pigmentation of its eyes, which are among the few parts of its body that are not transparent, has been specifically studied for evidence of adaptations to the bioluminescent light environment of the abyss. Its entire body is invisible. The only parts that are not are its eyes, and those eyes have been specifically redesigned to see in bioluminescent light rather than sunlight. The glass octopus is a pair of eyes that hunts in the dark, attached to a body that is not there. The whale fall ecosystem is one of the more remarkable phenomena of the abyssal zone, and one of the clearest illustrations of how biological productivity at the surface eventually reaches the deepest parts of the ocean floor in a form that sustains complex ecological communities. When a large whale stops functioning and its body sinks to the abyssal seafloor, it creates an energy bonanza in an environment where energy is almost unimaginably scarce. A single blue whale carcass contains an amount of organic material equivalent to what would otherwise rain down on that patch of seafloor over decades in the form of marine snow and other sinking particles. The ecosystem that assembles on a whale carcass on the abyssal floor passes through a series of successional stages that can last decades, beginning with mobile scavengers like sharks and hagfish that consume the soft tissue, proceeding through enrichment opportunists, organisms that colonize the bones and sediment to exploit the dissolved nutrients released by decomposition, and eventually arriving at a stage in which bacteria break down the lipid-rich bones through a chemosynthetic process that releases hydrogen sulfide, supporting a specialized community of organisms analogous to those found at hydrothermal vents. A single whale carcass can sustain this succession for 50 to 100 years. One body, one event, half a century of ecosystem. In a place where almost nothing happens, the arrival of a dead whale is the biggest thing that will occur in that square kilometer for longer than most human institutions last. The sediment of the abyssal floor is not mud, it is a record. The rate of sedimentation here is a few centimeters per 1,000 years, which means that a sediment core, 3 meters long, contains 30 million years of ocean history, layer by layer, season by season, preserved under the weight of 4 kilometers of water in conditions so stable that nothing disturbs it. Every organism that has ever lived in the surface ocean and stopped functioning eventually sank here. Its shell, its bone, its chemistry settled into this sediment and stayed. Climate scientists have pulled these cores to the surface and read in their chemistry the temperature of water that existed before humans, before mammals, before most of what now lives on land. The abyssal floor is the planet's long-term memory, and the organisms living in it are doing so inside an archive that stretches back further than anything alive today can comprehend. The abyss is not a single uniform environment. The thermal gradient across the zone, from 4,000 to 6,000 meters, is a fraction of a degree per 1,000 meters. differences that sound trivially small, but that are physiologically decisive for organisms whose entire biochemistry is calibrated to a specific temperature. An abyssal organism, transplanted just 1,000 meters up or down within the zone, would experience something analogous to a sustained fever or hypothermia, a disruption of enzymatic function that would compromise its ability to feed, reproduce, and compete. Every depth here has its own biology. The gradient is so finely divided that its inhabitants have been shaped by specific meters, not just the general category of the deep. The biodiversity of the abyssal plain was long assumed to be minimal, on the reasoning that an environment so low in food, so cold, so dark, and so physically uniform could support only a handful of highly specialized species adapted to extreme scarcity. This assumption was wrong, in the same way that many assumptions about the deep ocean have been wrong, which is to say, it was wrong in the direction of underestimating what was there. Systematic sampling of abyssal sediment using quantitative box cores, instruments that recover a defined volume of sediment from a specific location, has revealed a diversity of myofauna, the tiny organisms living between sediment particles, that rivals or exceeds the diversity of comparable habitats in the shallow ocean. Hundreds of species of nematodes, copepods, polychaetes, and other small invertebrates have been found in single samples from the abyssal plain, many of them new to science, and some of them representatives of previously unknown genera and families. The total number of species living in abyssal sediments globally, across all the deep ocean plains combined, has been estimated in the hundreds of thousands, a figure that would have been considered implausible before the systematic sampling work of the past few decades confirmed it. The abyssal plain is not a biological desert. It is a species-rich habitat whose biodiversity has been systematically underestimated because the organisms that live there are small, rare, and buried in sediment that is extraordinarily difficult and expensive to sample at the depths where they live. And the creatures that live even deeper, in the trenches that cut through the abyssal floor toward depths that the abyssal zone itself does not reach, have pushed the limits of what biology can do even further than anything found here. The manganese nodules that litter the abyssal plain across large areas of the Pacific, Atlantic, and Indian Oceans are among the most striking geological features of the abyssal zone and among the most economically contested. These potato-sized concretions of manganese, iron, cobalt, nickel, and other metals grow on the abyssal seafloor at rates of a few millimeters per million years. Among the slowest geological processes known, by the precipitation of dissolved metals from seawater onto small nuclei, typically a shark tooth or a fragment of bone. The nodule fields that exist across vast areas of the abyssal plain represent accumulations of billions of years of this slow growth, and the metals they contain are in high demand for battery technology and other industrial applications. The mining of abyssal manganese nodules has been proposed and partially piloted by several commercial entities, and the regulatory framework for abyssal mining in international waters is currently being developed by the International Seabed Authority. The ecological implications of mining operations in the abyssal zone are a subject of active scientific concern because the abyssal plain communities living on and among nodule fields are specifically adapted to the nodule environment, using the nodules as hard substrate for attachment in a region where hard substrate is otherwise absent, and the recovery timescale for disturbed abyssal communities, if they recover at all, is measured in centuries to millennia, given the extremely slow biological processes at these depths and temperatures. The conflict between the resource value of abyssal nodule fields and the ecological value of the communities living in them is one of the defining conservation questions of the deep sea in the coming decades. The recovery timescale for disturbed abyssal communities, if they recover at all, is measured in centuries to millennia. A single mining operation that crosses an abyssal nodule field could eliminate ecosystems that took millions of years to assemble in communities that will not return within any human planning horizon. The decisions will be made by people who have never seen a nodule field, about organisms that have never been named, in an environment that has never been fully mapped. The abyss will not resist. It has no mechanism for resistance. It will simply absorb whatever reaches it, the way it has absorbed everything else that has ever sunk to its floor, and hold it there in the dark for longer than civilization has existed. The currents that flow through the abyssal zone are slow, cold, and of enormous importance to the global climate system, and they connect the abyssal zone to the surface world in ways that the complete darkness and extreme depth do not suggest. The thermohaline circulation, the global system of ocean currents driven by differences in water density caused by temperature and salinity variations, has its deepest and coldest component in the abyssal zone, where the densest water in the ocean, formed at high latitudes by the cooling and sea ice formation processes that increase water density to its maximum, sinks to the abyssal floor and begins its 1,000-year journey through the ocean basins toward the regions where it will eventually be upwelled back toward the surface. This abyssal circulation connects every ocean basin and every depth, carrying dissolved oxygen from the surface to the deep, distributing nutrients from regions of upwelling to the abyssal sediment that depends on them, and representing one of the primary mechanisms by which the heat and chemical composition of the deep ocean are regulated over timescales of centuries to millennia. The abyssal zone is not isolated from the surface world by its depth. It is connected to it through the slow, cold rivers of thermohaline circulation that flow through it, and the organisms that live in it are, in ways they cannot experience or express, participants in a planetary circulation system whose full extent spans the entire global ocean. But connected as it is, the abyss is not the deepest place. Below 6,000 meters, the seafloor drops again into trenches that go deeper than any plane, where the conditions push past everything described here into something that biology should not be able to reach at all. The trenches that cut through the abyssal floor into the hadal zone begin at 6,000 meters and extend at their deepest to 11,000 meters in the Challenger Deep of the Mariana Trench. The word hadal comes from Hades, the Greek underworld, and the naming is more apt than most scientific terminology manages to be. The hadal zone is not simply more of the abyss at greater depth. It is a categorically different environment, defined not just by greater pressure and greater cold, but by its geometry. The trenches are narrow, elongated features where two tectonic plates collide and one slides beneath the other, creating a V-shaped valley in the seafloor that concentrates organic matter falling from above, channels currents in specific directions, and creates a physical isolation that has allowed the evolution of species found nowhere else. The pressure at the bottom of the Challenger Deep exceeds 1,000 atmospheres. The temperature is approximately 1 to 2 degrees Celsius. The darkness is as complete as anywhere in the ocean. And yet, when the first systematic biological surveys of hadal trenches were conducted using autonomous landers that descended to full trench depth, captured footage, and returned to the surface, what the footage showed was not the barren, lifeless environment that many biologists had expected. It was an environment teeming with life of a kind that should not, by any biological intuition formed at the surface, have been there at all. The hadal snailfish is the deepest living vertebrate known to science, and it holds the record at a depth exceeding 8,300 meters, set by footage captured in the Izu Ogasawara Trench in 2023. It does not look like something that should exist at 8,300 meters. It looks like a small, pale, almost translucent fish, a few centimeters long, moving across the trench floor with what appears to be an unhurried confidence in an environment that would crush an unprotected human body instantly. The snailfish's biochemistry is the reason for its presence at this depth, and the specific molecular adaptation that allows it to survive where nothing else vertebrate can reaches its physical limit somewhere between 8,000 and 8,500 meters depth, constrained by the solubility of trimethylamine oxide, the compound that stabilizes its proteins against the pressure-induced structural changes that would destroy the function of any surface organism's proteins at these depths. Trimethylamine oxide accumulates in the tissues of deep-sea fish in concentrations that increase with depth, counteracting the pressure that would otherwise distort protein shape and halt enzymatic function. But trimethylamine oxide itself has a maximum solubility in biological tissue, and at the pressures corresponding to approximately 8,000 to 8,500 meters, that maximum is reached. Below that depth, the compound cannot accumulate in sufficient concentration to protect the proteins any further, and the biochemical solution that has allowed snailfish to colonize the hadal zone runs up against a hard physical limit. Whether any vertebrate has found an alternative solution to this limit, some different molecular strategy that works at the pressures of the deepest trenches, is not known. The Challenger Deep, at 11,000 meters, sits more than 2,000 meters below the deepest confirmed snailfish record. What lives there, if anything vertebrate lives there, has not been documented. The invertebrates of the hadal zone face the same pressure constraints as the vertebrates, but have had more evolutionary time across a wider range of body plans to find solutions to them. And the result is a community of hadal invertebrates that is both more diverse and more extreme in its adaptations than the vertebrate community. The amphipods, small crustaceans related to sand fleas, are among the most abundant organisms in hadal trenches, and have been found at the full depth of the Challenger Deep, making them among the deepest living animals ever confirmed by direct observation. Hadal amphipods can reach sizes of 30 centimeters or more, substantially larger than their shallow water relatives, a phenomenon known as hadal gigantism that appears to reflect the specific combination of cold temperature, high pressure, high food availability, and low predation pressure that characterizes the trench environment. The cold slows metabolic rate, which in combination with the abundant food available in the concentrated organic matter that accumulates in the trench, allows individuals to grow larger before reproductive investment becomes more efficient than continued growth. The low predation pressure means that large size is not penalized by increased visibility to predators in the way it might be in shallower water. Hadal amphipods are therefore very large by the standards of their relatives, and they are among the most voracious scavengers in the deep sea, capable of stripping a large fish carcass to bones in a matter of hours when they aggregate in the food-concentrated environment of the trench floor. The discovery of plastic pollution in the Hadal zone is among the more dispiriting findings of deep-sea exploration in recent decades, and the concentrations found in Hadal trenches are, in some cases, higher than those found at the surface of some of the most polluted ocean regions. This is not coincidental. The trenches function as natural collection points for material sinking through the water column, concentrating not only organic matter, but any persistent material that reaches the ocean surface. Plastic particles, which are produced in quantities measured in hundreds of millions of tons annually, and which are resistant to the biological and chemical degradation that breaks down organic material, have been accumulating in the ocean for decades and have now sunk in the form of microplastic particles to the deepest places on Earth. Sampling of Hadal Trench sediments has found microplastic concentrations that exceed those in surface ocean garbage patches, representing the accumulation of plastic over decades in environments that have no mechanism for breaking it down and that are too remote and too deep to be practically cleaned. The Hadal amphipods themselves have been found to contain plastic in their digestive systems, ingesting microplastic particles along with the organic matter they consume. The deepest environment on Earth, the environment furthest from any human settlement or any human activity, contains clear chemical evidence of industrial civilization in the tissue of animals that have never been exposed to the surface world. The trench goes all the way down. The plastic got there first. The hydrothermal vents that occur in some Hadal trenches, where geothermal heat and chemically modified water enter the trench environment from below, create biological hotspots within an already extreme environment. The discovery of vent communities at Hadal depths, supporting the same chemosynthetic base of bacteria that powers vent communities at shallower depths, extended the known range of vent ecosystems to environments previously assumed to be too extreme for even the most pressure-adapted chemosynthetic organisms. The vent communities in Hadal trenches are less diverse than those at shallower vents. Constrained by the combination of high pressure and the physical isolation of the trench environment that limits the dispersal of species from other vent sites, but their existence demonstrates that chemosynthetic productivity operates at the full depth range of the ocean. The deepest trench in the ocean contains ecosystems that do not require the sun at all, not even indirectly through the sinking of surface-produced organic matter. They run on geothermal chemistry, on the heat and reduced compounds rising from the tectonic processes that created the trench in an environment so isolated from the surface world that it might as well be on a different planet. The question of why the hadal zone contains as much life as it does, given the extreme conditions that characterize it, has generated a body of ecological research that has replaced the original assumption of hadal sterility with a more nuanced picture. The key is the same geometry that makes the trench extreme. The V-shape of a trench, narrowing with depth, is an efficient collector of organic matter sinking from above. Particles that might disperse across thousands of square kilometers of abyssal plain, if they sank in open water, are instead funneled by the trench walls toward the narrow floor, concentrating food at the bottom in quantities per unit area that substantially exceed the concentrations found on the adjacent abyssal plain. The trench floor is, counterintuitively, a food-rich environment relative to the abyssal floor surrounding it, and this food richness supports higher densities of organisms than the surrounding plain, despite the more extreme physical conditions. The extreme conditions filter out everything that cannot tolerate the additional pressure, leaving a community of specialists with access to a more concentrated food supply than anything in the surrounding abyss. The deepest place on Earth is, counterintuitively, one of the better fed places on the ocean floor. The deeper you go, the more paradoxical the rules become. The biochemistry of hadal organisms, beyond the pressure adaptations of their structural proteins, contains surprises that connect to some of the most active areas of biotechnology research. Enzymes isolated from hadal bacteria and other hadal microorganisms have been found to retain their catalytic activity at pressures and temperatures that would destroy the equivalent enzymes from surface organisms, making them potentially valuable as catalysts for industrial processes that require enzymatic activity under conditions that surface organism enzymes cannot tolerate. The class of enzymes called cold-active enzymes, which catalyze biochemical reactions efficiently at low temperatures where surface organism enzymes are too slow to be useful, has attracted pharmaceutical and industrial interest for applications ranging from food processing to pharmaceutical synthesis. The hadal zone has produced molecular tools that human technology has not independently developed, and that the surface world's biology would not have produced because the selective pressure that generates them does not exist above the pressure. Hadal depth range. The deepest organisms on Earth are inadvertently producing chemistry that the most sophisticated biotechnology industry on the planet finds useful, in a place that industry has never visited and may never visit. The acoustic environment of the hadal trench is one of the least studied aspects of these ecosystems, and what little monitoring has been done has produced results that are not easily explained. Hydrophones deployed in the Mariana Trench have recorded sounds that include the low-frequency vocalizations of baleen whales traveling thousands of kilometers away, the sounds of earthquakes and distant ships, and sounds whose sources are not readily identifiable. The trench acts as a natural acoustic conduit, channeling sounds that enter from above and propagating them along the trench floor in ways that the local topography amplifies and distorts. What organisms in the hadal trench are doing with these sounds, whether any of the acoustic information reaching the trench floor is detected and used by the trench community is not known. The trench is full of sound. What is listening to it is still a question science has not been able to answer because science has barely been there long enough to ask it. The geological dynamism of the hadal environment means that the hadal zone is not a static environment in the way that the abyssal plain largely is. Earthquakes that originate along the subduction zone where the trench is forming send tremors through the trench floor and walls, disturbing the sediment and occasionally triggering turbidity currents, avalanches of sediment and organic matter that cascade down the trench walls and deposit concentrated organic material on the trench floor. These disturbance events create pulses of food availability that the hadal community has adapted to exploit, with the scavenger communities capable of mobilizing rapidly in response to a large organic input event. The trench is not merely a place where extreme conditions persist indefinitely. It is a place where those extreme conditions interact with ongoing geological processes that periodically reshape the environment and redistribute the resources that the community depends on. The deepest place on Earth, the Challenger, deep in the Mariana Trench, at approximately 11,000 meters, has been visited by human beings exactly 3 times in the form of crewed dives, twice in the 1960s by the Trieste, and once in 2012 by James Cameron in the Deep-Sea Challenger. The total time that human beings have spent at the very bottom of the deepest trench on Earth is measured in hours. Everything that is known about the biology, chemistry, and geology of the Challenger deep floor has been learned from autonomous landers, remotely operated instruments, and the analysis of sediment samples returned to the surface. The deepest point on Earth has been seen by human eyes for a few hours over the course of 60 years, and those hours were separated by 52 years. Whatever is at the bottom of the Challenger Deep right now, whatever organisms are moving across the sediment, and whatever chemistry is occurring in the vent fluids, and whatever sounds are propagating through the trench at this moment, is happening without any human witness and without any instrument currently in place to record it. What does it mean that the deepest place on this planet is also one of the least visited places on this planet? It means that the end point of the journey described here, the bottom of the bottom, is not a place that science has characterized and moved on from. It is a place that science has barely touched. The species found in the Challenger Deep have been sampled at a handful of sites along a trench that stretches for hundreds of kilometers. The chemistry of the vent fluids has been measured in a small number of locations. The sediment record has been cored at a few points. The acoustic environment has been monitored intermittently. There's no permanent monitoring station, no standing scientific presence, no continuous record of what is happening in the deepest place on Earth. The biology there is being shaped by processes that nobody is watching, producing adaptations that nobody is documenting, generating chemistry that nobody is sampling. The deepest organisms on Earth are doing what they have been doing for millions of years, in a place that the species that named them has visited for a total of a few hours. The journey ends here. But what lives at the bottom of this journey has barely been introduced. The deepest you can go on this planet is also the place you are least likely to ever go, the place that has been visited less than the surface of the moon, the place that every layer of the ocean above it has been preparing you to understand. The journey down through 200 meters of familiar light, through the darkness where the hatchetfish solve differential equations with their own bodies, through the absolute black where anglerfish dissolve their mates into themselves, through the abyss where single cells build cathedrals from sediment, ends here, at the bottom of a trench that is still adding to its geology, while everything above it slowly sinks through the dark. The deeper you go, the more impossible the biology becomes. And at the very bottom, what lives there is not impossible anymore. It is just unknown. If this journey moved something in you, consider subscribing to Sleep on Deep. Every week, we descend a little further. The ocean is waiting.